Welcome to Unit 3, and in this unit, you'll learn everything you need to know for cellular energetics.
The big idea here today is we're going to talk about where it occurs, and it occurs in the cytoplasm of cells. And it occurs in almost all cells. That includes bacteria, humans, plants, animals, all of it. Now, what happens is it's enzyme catalyzed steps to start breaking glucose down into two pyruvate molecules. We then use this energy, this initial energy of breaking this molecule down to create some ATP and also NADH. The kind of question I want you to think about today while we're going through this lesson is you need to ask yourself, why is glycolysis, why is it universal? Because it's found in almost all cells. That includes prokaryotes, archaea, and also eukarya, which includes animals and plants. Okay, most of its steps happens in the cytoplasm, so there is no specialized organelle needed for the process of glycolysis to occur. And it begins the energy transfer from glucose, and then we have various enzyme controlled steps to ultimately break glucose down into pyruvates. And what we get out of this chemical reaction is we get some ATP that's been created and also NADH that carries these electrons. Now, the essential knowledge is that glycolysis starts the energy transfer. It is the first step of breaking down a 6-carbon molecule into pyruvate. Okay, and pyruvate is a 3-carbon molecule and we'll have 2 of those molecules. So no carbon is lost. From this reaction, we create a little bit of ATP with substrate level phosphorylation. Okay, so it's not using the electron transport chain to create that ATP. And because we are breaking these chemical bonds, these are the chemical bonds, and because we're breaking these chemical bonds to rearrange the carbon, we have electrons released. And those electrons reduce NADH. Glycolysis happens within the cytoplasm and it occurs in almost all cells. Okay? It does not require oxygen. So oxygen is not required. It can happen with oxygen, it can happen without oxygen, but it's important that we are able to regenerate NADH back into NAD plus so that glycolysis can continue. And then finally, this is a conserved pathway, so we find it in bacteria, archaea, and eukarya. It is shared across life. So this is across life how cells start the breakdown of glucose. Now, just to make sure we understand glycolysis, it does not require oxygen directly, okay? What it needs is that it needs to be able to recycle this NADH molecule, either by sending it to the electron transport chain, where it's reduced and it can return back to glycolysis, or with fermentation, meaning we are using a process called fermentation to now recycle these molecules so they can be sent back to glycolysis. Okay, because ultimately this is what we need to recycle. We need to get rid of these electrons so that it can accept electrons again from breaking down glucose. It does not release carbon dioxide, meaning it starts out with 6 carbons, and I end up with 2 molecules that have 3 carbons each. Okay, so no carbons have been lost. They've only been rearranged. And then glycolysis does not produce the most ATP. The most ATP from respiration comes from the electron transport chain that creates the gradient and then chemiosmosis. This is where most of the ATP comes from. Glycolysis creates very, very little ATP. It creates 2 net ATP. And glycolysis is not the entire process. It is only the first step. If you like that lesson, don't forget to like and subscribe, and I'll see you for the next one.